this seminar. I will introduce the speaker today. His name is Lopez Valdivia. He is a PhD student in Penn State University in the group of Jonathan Lynch. Maybe some of you have heard about his job in Roots. And Ivan is the last year of his PhD. He's here to do some field work in Obregon, but he wanted also to show us his job in roots in maize and wheat, and he's more specifically studying the phenotypic landscape of roots, mainly in maize with applications in wheat. So let's welcome Ivan, and please.
Hello. This is not working, right? Thank you all for being here. My name is Ivan Lopez, and I will present my research on maize and with some applications on wheat as well. All the principles that I will present here are applicable on wheat. And well, thank you for being here, and thank you for the people in Zoom for connecting. All right, and we will go further here. So I would like to use this one so I can. Hello. This is working, right? Okay. So first of all, okay, so this is a summary of the talk I'm going to give today. First of all, we're going to talk about what are the Phenes, what are the integrated phenotypes, what is a fitness landscape, and what is the model OpenCIMMYT. After that, I'm going to give an example of how we study the fitness landscape of maize, specifically talking about the cortical cell size and cortical cell file number. And after that, we're going to discuss what are we doing in terms of the role of MCS, which is a new phenotype in maize, and the role of the domestication of maize. So we're going to talk about the first topic now. So what are the Phenes? So we say that Phenes are the most elemental components of the phenotype. And to illustrate this point, I'm going to talk about the root diameter. For example, if you go to the field, you sample a root, you can measure the root diameter. But if you look closer, there is other anatomical components that give rise to this root diameter. For example, we know that we have the stele, which is the central cylinder of the root, and we have the cortex. And in the cortex, for example, we have the cortical cell size, and we have the number of files in the cortex. And these two traits in combination, they're going to give the total root diameter. So the concept here that I want to just make an example of is that there are many smaller components of the phenotype that give rise to this bigger phenotype, which is root diameter. And this is just an example. This can be applied also for architectural root traits, such as number of nodal roots or other traits. And for example, all these Phenes interact with each other to affect nutrient acquisition, nutrient utilization, or carbon economy. And at the end, this interaction between Phenes affect yield. And just to give another example here, for example, we have six different environments here. We have low nitrogen and we have high nitrogen. And we have two Phenes that we are testing. This is the first Phene, ground root number, and we have the Phene states, which is the possible distribution of ground root number in maize. And we also have the root cortical arrhythmia, which is another Phene. And we also have the Phene states, which are the distribution for this Phene. And we can see that under high nitrogen conditions, optimal conditions, having a higher number of roots and any number of root cortical arrhythmia is good, is positive, or is beneficial for the plant. However, if we change to a condition where we have low nitrogen, now we see that having a less number of ground roots is better, and also having increased root cortical arrhythmia is better. So this is telling us that the combination of Phenes is going to affect the plant performance in different environments. And these are just two Phenes. I am just showing the ground root number, but we have seen different Phene states for other Phenes, which are, for example, root cortical arrhythmia we see that the variation goes from 5% to 30%. Also, the number of cortical cell file number goes from 8 to 20, and the cortical cell size goes from 100 to 500 micrometers. For the lateral branching density now, looking at the architecture of the roots, we know that there is a phenotypic variation for lateral roots in terms of 2 to 12 branches of roots per centimeter. And also we have variation for nodal root number, and we have variation for angle. So what I want to talk about here is, if we consider all the possible combinations of Phenes states that in total give rise an integrated phenotype, the number of combinations is really high, and it's hard to test this in the field or in the greenhouse. Fortunately, we have a model, which is OpenSIMMYT, which simulates what will be the root architecture and the root anatomy of a specific root system, and we can vary different Phenes states. For example, we can vary the branching density of the roots, we can vary the number of nodal roots, we can vary the angles, and we can vary the anatomical root traits in terms of construction cost and root maintenance. And we're going to talk more about that. So for example, this is just how the roots are simulated. And for example, here the red color, well, the gradient of color is the nitrogen leaching. And yeah, well, just to show how we have used this model in the last years. For example, in 2011, the model predicted that having higher cortical parenchyma was beneficial for the plant under low nitrogen conditions. Then in 2014, we took this to the field, and then we observed that in the greenhouse in South Africa and Pennsylvania, the lines of maize that had higher parenchyma performed better or had higher yields. Also for lateral branching density, the model predicted in 2014 that having a less number of branches per centimeter is going to be better for capture of nitrogen. And then we took this, well, they took this into the field, and they observed that indeed the lines that had fewer lateral branching density, they perform better under low nitrogen conditions. And another example, it was tested in the model that under polyculture, the root was able to uptake nutrient, nitrogen more efficiently. This was tested in the model. The model predicted that in polyculture, the uptake of nitrogen was more efficient compared with monoculture. And then in 2014, this was tested in the field, and it was congruent with the model. So this is just like an introduction of how the model has helped us to guide research in discovering new Phenes. And most of the times, or in many examples, the predictions of the model have helped in finding these root traits. Now we're gonna talk more about this fitness landscape of roots, specifically focusing on cortical cell size and cortical cell file number. And this is the work that I have been doing in Penn State. So we came to this hypothesis because in 2014, Chimungu showed that having less cortical cell file number reduced the respiration per centimeter of root segment. And since there is less respiration, the roots were able to grow longer roots and grow more tissues. And whenever they test this into the field, they observe that under drought, the lines that had a reduced cortical cell file number, they perform better. They produce higher yields. So also they test for cortical cell size, and they observe that the larger the cortical cell size, the less the respiration, and the effect was the same in the field. Whenever they took these plants into the field, they observed that the plants that had larger cortical cell size, in this case, well, there is large cortical cell size, they develop higher yields under drought. So water stress is white. So you can see here under water stress, there is a higher effect. So what's happening here is that because there is less respiration, less carbon was used for maintenance of the tissue, and that carbon can be used for developing root structures instead. So the roots were able to grow deeper into the ground and capture this water. This was true for water stress. So we have the hypothesis that since nitrogen is a mobile resource, it will have a similar effect. So we grow 12 different genotypes in the greenhouse under high and low nitrogen conditions. And we measure this first, the cortical sulfide number, and we observe variation. We have some genotypes that have a smaller cortical sulfide number, and some other genotypes that have a higher cortical sulfide number. And then in order to know the construction cost and the maintenance cost of the root, we measure respiration. So respiration is telling us what's the maintenance cost of the roots in terms of carbon, and we also measure nitrogen content. And nitrogen content is telling us the construction cost of the tissue in terms of nitrogen. So we observe a similar pattern that Chimungo observed before, that the lines that have a reduced cortical sulfide number, they tend to have less respiration, and they tend to have less nitrogen content in the tissue. So we wanted to test if the lines that had a large or higher cortical sulfide number or a smaller cortical sulfide number were better under nitrogen or were adaptive under nitrogen. But we had a problem that sometimes the lines that had, for example, this one that have increased cortical sulfide number, sometimes it has different values for cortical cell size. And since I showed before, cortical cell size is also affecting the respiration of the tissues. So in order to test these independently, like only the values for cortical sulfide number, keeping constant the cortical cell size, we developed, oh, sorry, well, I forgot to show them. We did the same for cortical cell size here. We also measure respiration, and we also measure nutrient content. And coming back to this part of testing independently those values, we built a multiple regression model using cortical sulfide number and cortical cell size as response variable, as predictors, and the respiration as the response variable. So basically, we are controlling, like coming from the same data that we collect in the greenhouse, we are just making this multiple regression model to explore a decision space in which we can say we have a constant number for cortical sulfide number, and let's vary the number of cortical cell size and see what will be the respiration, or what will be the nitrogen content, or the root diameter. And this is how if we plot the models, this is how they look. Basically, for respiration, if you have larger cortical cell size, you have less respiration. And if you have a smaller number of cortical sulfide number, you also have less respiration. So we use the model to predict an example of what will be a reduced and increased cortical cell file number. In this case, 6 cortical cell files and 13 cortical cell files. And this is how it looks if we build the 3D architecture of the root. First of all, in color red, we see how the nitrogen is leaching, and the changing color represents the concentration of nitrogen. And at first glance, we can see that the root systems that have a reduced cortical cell file number, they tend to reach deeper layers of the soil in the model. So this is what I was talking about before. For example, these are the values that we use for cortical cell file number, 6 and 18. And then we keep the cortical cell size constant in 360. And these are the predictions of the model that come from the multiple regression model from the data from the greenhouse. So at first glance, we can see that there is less respiration when you have decreased cortical cell file number, and you also have less nitrogen content. So in terms of maintenance of the tissue, we have less cost of maintenance and less cost of construction cost. And whenever we plot this against, for example, here we are showing the x-axis. I'm showing what will be the level of stress. 100 represents 0 stress, and 0 represents the higher stress. And here is the comparison of the decreased cortical cell file number versus increased cortical cell file number. So here, basically, we are showing whenever you don't have a stress, there is no difference between the reduced or increased cortical cell file number. But once you start increasing the stress, the plant that has a decreased cortical cell file number improve like until 80% of the biomass in the model. And since the good part of using models is that we can test each part independently. So we say, okay, for example, what will happen if we have only the effect of respiration? So we keep constant the values for root construction cost in terms of nitrogen, and we use the value for respiration. And we observe that the contribution of respiration itself is around 30%. We did the same, but now for nutrient content, and we observed that the benefit was also around 30%. And whenever we test the effect of diameter by itself, we observe that the diameter is actually giving a benefit of 35% or almost 40%. So this was interesting because there is a relation between cortical cell size and diameter, because whenever you increase the number of files, you also increase the diameter, which also increases the cost of maintenance of the tissue. So whenever you have a smaller, a reduced cortical cell file number, you have less diameter, and the diameter is also helping the plant. So that was a good finding that we observed for the cortical cell file number, and we did the same for cortical cell size. So here we also simulate what will be the phenotype of small cortical cell size and large cortical cell size. We run the models, we build the animations, and in this animation, I just want to exemplify how the model is able to simulate in every segment of the root what will be the nutrient uptake. For example, here we just make a close-up here, and we can use, I just want to show you how for every segment, every small segment of the root, we simulate the nutrient uptake. So it's a quite complex model that takes considerable time to run. But anyway, coming back to the data, again, these are the predictions. So now we vary the cortical cell size. So now we have 450 is a large cortical cell size, and 200 will be the small cortical cell size. And we keep constant the cortical cell file number. And same than before, we obtain less respiration and less nutrient content in the plants that have large cortical cell size. And whenever we evaluate the benefit of these plants in the model, we observe that under high stress, we will obtain until 25% benefit. So doing the same for respiration, we observe around 15% of benefit, and for nutrient content, around 5% of benefit. However, for diameter in this case, when you increase the size of the cortical cells, you also increase the diameter. And as I was discussing before, increasing the diameter of the root increases the cost in general, and will not help the plant in a stress condition. So whenever we look at the effect of diameter, for cortical cell size, we see that the effect of the diameter is actually not helping the plant. It's a negative value. But again, since we are using models, we can use these artificial phenotypes. And we say, okay, what if we found a genotype that has a large cortical cell size but keeps the root diameter small somehow? So we did this transphenic phenotype, which is basically having the benefits of respiration, of nutrient content, but also having a small diameter. And we observe that it has a benefit until 30%. So, but here, okay, this is the thing. Here we are just observing only large or small cortical cell size. We are not looking, at all these possible combinations that I was talking at the beginning. But since we have the multiple regression model and we have the open SIMMYT model, we can test every possible combination. So I just took what will be the respiration and nutrient content of different levels of cortical cell file number and different levels of cortical cell size. And I established 3 different environments, which the only difference is the level of nitrogen we use. So here we use low stress, which is like 25% reduction of the optimal conditions. And here we have high stress, which is 75% reduction in the of the potential growth of the plant. So one thing that we can realize here, if we look at low stress conditions, is that the effect of cortical cell size is important only when you have increased cortical cell file number. For example, here, if we look at this row only, we can see that, okay, having a large cortical cell size is improving the shoot biomass, right? And having a small cortical cell size is affecting the biomass. But if we look at the scenario whenever we just have 6 file number, we see that the effect of cortical cell size is not relevant at all. So this means that under low nitrogen stress, the effect of cortical cell size is only important whenever we have a number of files that goes beyond 12. However, if we look at the high stress, we see that the importance of both rates is, or both rates are important in all the conditions. For example, here, even though you have a reduced cortical cell file number, which before we test that it was beneficial, it's not enough for the plant to perform the best shoot biomass. So you need to combine the small cortical cell file with a large cortical cell size, and then you have the best performance. And again, just to emphasize that in the model is an artificial way in which we can test this fitness landscape, which will be how different Phenes states could perform in different environments. And then since we add another variable, so now we have the same combination, we have different levels for cortical cell file number and cortical cell size, but now we say, okay, what will be the effect of the combination of these 2 traits, but with the addition of cortical arrhythmia, which before root cortical arrhythmia was tested, and it was proved that was improving plant performance under low nitrogen conditions. So, and I test this under 2 different environments. Now we are showing here seed loam soil and loamy sand. So now the nitrogen concentration is the same, but the stress is coming from the soil texture, not from the concentration of nitrogen. And we see that in general, when you have a loamy sand soil, you have more stress plants. For example, here the ranges in seed loam go beyond, go between 10 and 14 grams of biomass. And in sandy soil, we have from 3 grams to 5 grams. So in this scenario, the plants are more stressed because of the soil texture. And we see a similar pattern whenever the importance of cortical cell size, which is represented here in the y-axis, is relevant whenever you have more stress conditions. For example, in this scenario, when you have sandy soils. When you have sandy soils, in any value of cortical cell file number, having a large cortical cell size will be better. You know, in this plot, in these 3 plots, is the same scenario. But if you have a seed loam soil, when the plant feels less stress, it doesn't really matter what's your cortical cell size if you have a reduced number of cortical cell file number. But if you have an increased cortical cell file number, then your cortical cell size will be relevant. So, and with this, we just make the conclusions that we show quantitative evidence that large cortical cell size and reduced cortical cell file number are adaptive phens that useful for capture of nitrogen under low nitrogen conditions. And the benefits of these 2 traits is associated with the nutrient availability, the soil type, and interactions between phens. And then we test that a combination of larger cortical cell size, reduced cortical cell file number, and higher enchyma will be appropriate or useful under sandy loam soils. So now we can jump into the the third part of the talk, which is exploring the fitness landscape of MCS in the domestication of maize in silico. So first of all, what is MCS? MCS is a phenotype that was discovered in 2021 by Hannah Schneider. And basically, if you take a segment of the root and you look at the other cortical cells, when you have MCS, which is a multiseriate cortical sclerenchyma, you will see that there is an increase of lignin in the cell walls. And this increase of lignin is reducing the cell size. So as we talked before, reducing the cell size is beneficial for metabolic processes, but also this increase of lignin is increasing the strength of the root, the tensile strength of the root. So in 2021, they test what will be the case if they test maize lines with and without MCS under compacted soil conditions. For example, here we have compacted soils, non-compacted soils. And first of all, they observe that the roots from MCS lines, which are in blue, they develop deeper root systems. And under compacted soils, they'll perform a higher biomass in two different fields. So MCS is a cortical trait that increases the strength of the root and increases the plant performance under compacted soil conditions. But interestingly, also in 2021, they screened several genotypes of wild varieties of maize, also of wild varieties of wheat and barley, and they observed that MCS was not present in any of those. So MCS was present only in modern varieties, suggesting that maybe some change in the domestication of maize produced that MCS was useful for something. But in last year, well, this is a work that we did also in Penn State. We have these roots from 5,000 years ago. These roots they were found in the Tehuacan Valley in Puebla. And so we analyzed the root architecture and the root anatomy of these ancient roots, and we observed that MCS was present there. So it's quite interesting because it's telling us that 5,000 years ago, this root was fixed for some reason, but we don't understand exactly how. And trying to go further on this, trying to understand what were the agricultural conditions in the Tehuacan Valley 5,000 years ago, we found this research of these archaeologists, and they show that it was a very complex irrigation system that was built between 8,000 to 6,000 years ago. So we can think in the agriculture in the Tehuacan Valley 8,000 to 6,000 years ago, and they had a huge system of canals that were irrigating the crops. So, okay, we can think that water then was not the stressful factor, even though the Tehuacan Valley is so dry. Water was not the constraint of these plants because they had these irrigation systems. And it's important to mention that in the Tehuacan Valley, they have empty soils, which are a very hard soils. And just to illustrate the successful of this irrigation system, this is a work from McNish in 1987, and they found like several specimens of avocado and black zapote. And those plants are tropical plants. You need a lot of water to grow these plants. So this is just an example of how successful the irrigation systems were in the Tehuacan Valley. And looking, like going further in what will be the ancient environments in terms of agriculture, we found this article also from some archaeologists. And they show that across time, for example, here we have 10,000 years ago, and here we have present. But they measure what will be the isotopic nitrogen in the tissue of several paleobotanical specimens, like going from the oldest ones to the newer ones. And they observe that the concentration of this nitrogen was smaller in the modern specimens, suggesting that, so they make this relation between the fertility of the soil with the amount of nitrogen in the tissue. So they conclude that the fertility of the soil was getting lost across time. But also they show that in those environments, they were increasing the yields. So it was quite interesting because this means that across time, they were increasing the yields or increasing the seed size of wheat and barley in this case, but in a soil that has less nutrients across time. So, and just adding more evidences into this idea, this is a work that was published in 2022 that tested what will be the effect of increase in penetration ability of roots in soils that are compacted and also low nitrogen, which is a scenario in Tehuacan Valley, right? We just show evidence that the Tehuacan Valley had compacted soils, but also probably low nitrogen conditions. So root systems that had an increased penetration ability perform better under low nitrogen and compacted soil. So here we are showing, for example, this one gradient represents a soil with homogeneous compaction. Flow pan represents a soil with a hard layer. Well, flow pan, we will focus here in flow pan, which is the one that has the hard layer. So we observe that having a high penetration ability, which is 400, 4,000 and 1,000, or 4,000, 4,000. In these two cases, having high penetration ability improve the performance of those plants. So just to make a summary of the evidences here, we know that MCS improves plant performance under compacted soils. We also know that MCS is absent in wild relatives of maize, but was present 5,000 years ago. We know that the agroecosystems in Tehuacan Valley used irrigation, and they had hard soils 5,000 years ago, and they still have hard soils. And agricultural evidence from Mesopotamia tell us that across time, we have less soil fertility. And root systems that have higher penetration in axial roots improve plant performance in compacted soil conditions. So all these evidences, we are just going to put them together to test if the MCS of the ancient root 5,000 years ago was actually important for the establishment of Mesoamerican agriculture or agriculture in the Tehuacan Valley. So the good part is that we have the phenotype, the architecture phenotype of the ancient root, and we can recreate what will be the root architecture of this root system in the model OpenCIMMYT. And we also have another model which is called root slice that was recently published this year, that from the anatomy, we can estimate what will be the root maintenance and the root construction costs that, as we talked before, root maintenance and root construction costs is important for plant performance. So we can simulate both. And since OpenCIMMYT also have a soil model and an atmosphere model, we can simulate what will be the agricultural environment, like modern, future, and past agricultural environments. And then we can test if this ancient root was actually doing something, or if MCS was relevant for the establishment of agriculture in Mesoamerica. So here is just some preliminary data related to that. It's just, we're just showing here that so cell to wall lumen ratio is basically the presence of parenchyma. So if you have, the presence of MCS. So if you have a value that is higher than 2, you have MCS. And here we're just showing that some maize lines that have MCS, they also tend to have higher stomatal conductance and less respiration. So, and with this, I hope to share more results in the next couple of months or years. And with this, I just want to conclude my talk. Thank you so much. And I will be open for questions.
Does anybody has a question here in the room first?
According to your data, they might give us light about the nitrogen management under different soils, texture, and what you mentioned that it must be a balance between the nitrogen and the water content in the soil. And finally, if you can tell us how you define reduce nitrogen regime. Could be 0, 50, 100, or what do you consider reduce?
Yeah, that's a good question. Thank you. So we define the nitrogen regime based on well, there are different factors in the model. For example, we have the optimum nitrogen and minimum nitrogen content in the tissue. So that is going to tell us how much nitrogen the plant needs per gram. So that's one factor there. The other factor is the nutrient concentration in the soil. So for that, we just represent like what will be the like a high fertilization in a field in terms of nitrogen per hectare, like grams of nitrate per hectare. And then we're going to start reducing the amount of, well, the concentration of nitrogen in the model. And this relation between the maximum and minimum nutrient content in the tissue will determine the stress. So there is a simple operation that is subtracting you have the uptake of nitrate minus the optimum divided by the optimum minus the minimum nitrate. So with that formula, you're going to calculate the stress factor. So the stress factor is going to reduce the growth rates of the shoot, and then the growth rates of the shoot are going to decrease the carbon input, and then the carbon input is going to decrease the growth of the root and the growth of the shoots as well. But yeah, in general sense, I mean, we take a concentration from the field, like what will be the higher amount of nitrate applying to the field. Then we represent that in the model as a concentration that the plant will be able to uptake or not, and depending on the uptake is going to determine the stress factor of the plant.
Usually, as breeders, they recommend us to look for a higher biomass, root biomass. How your results will help us to define what is the optimum root biomass?
That's a really good question. And for example, I will say that depends on the environment, because for example, you could have a high resource environment or a high input environment in which all the nutrients are there or the water is there. So you don't need many roots. And the roots are actually, the roots are not photosynthetic tissues. The roots are heterotrophic tissues that are always like consuming carbon. So having more roots in that environment is not helping at all the plant, because the roots are representing a waste of resources. If you have enough nitrogen and water, but if you don't have enough nitrogen and water, then we can see different strategies, because in a low input environment, we could have different scenarios. We could have maybe a low nitrogen environment or a low phosphorus environment. And those are very different, because in a low phosphorus environment, the limited of the resources are in the top of the ground or of the soil. So we need root systems that explore more efficiently the top of the soil. But if we are having a low nitrogen environment, maybe finding a root phenotype that reach deeper layers of the soil or deeper domains of the soil will be more efficient. That's why this idea of the fitness landscape of roots is like, what will be the effect of multiple combination of root fins under different environments, and test what will be the optimum root phenotype for each environment. So I will say there is not like a particular environment, sorry, a particular phenotype, but depends on the environment you grow them. Yeah, for drought, we have an idiotype that is the steep chip and dip, which basically proposes to have deeper roots, which are like steeper angles, but also anatomical fins that reduce the metabolic cost and therefore allow the plants to develop higher, well, to use the carbon more efficiently and then develop deeper roots or longer root systems. That's another good question, because so this idea of the fins, because for example, most of the times if you look at big phenotype and you try to do a QTL analysis or a GWAS, if the phenotypes are big, the phenotypes are quite complex, then whenever you target your genes or your locus, it will be hard to find. That's with complex phenotypes. But if we dissect the phenotype in a way that we can say, okay, maybe the root mass is actually the sum of all these smaller fins, and then we can do the GWAS for all those smaller fins, and then we can say, okay, maybe if we select for those instead of the whole root mass, we can find a better genetic association. But there is also plasticity, you know. So there is also plasticity, and if these fins are plastic somehow, maybe even though you select that, I mean, there is like several levels of complexity.
Yeah, there is a lot of variation, no? But you're right, according to the environment, you have to select.
Yeah.
Thank you.
Yeah, thank you.
Any other question?
Just wondering about the genetic variation. Have you looked kind of in a diversity set if your concept is correct of the size and the number of files?
Yes.
And correlated to actual phenotypes?
Yeah, you mean like in a diversity panel of, yeah. So we have this side project. Well, it's not a side project. It's actually my second chapter of the dissertation that is based on local adaptation of root traits. So we are basically exploring, well, in 2013, I think, Burton published an article in which she quantified the root architecture and the root anatomy of 195 land races and 60 teosintles. And so we have the diversity of phenotypes available. So we are planning to do a functional analysis with the models, because in those days we didn't have these capacities to simulate the anatomy and the root architecture. So we are planning to simulate all those 195 phenotypes for the land races and test basically if the ones, well, that's the approach. Testing if the ones coming from arid regions, they have phenotypes that help them to adapt to those arid regions, and same with the humid areas. So we found some interesting patterns that the land races from arid regions, they tend to have low number of crown roots, which is beneficial under drought. And there are also different plastic responses there because, well, yeah, there is a whole thing there because we grow those plants under drought and normal conditions. And we observe that the phenotypes from humid areas are more plastic, but not necessarily better, because what they do is the humid land races, they reduce the number of crown roots. But the land races from arid regions, they grow since the beginning and they have a reduced number of crown roots. So they are less plastic, but maybe more successful because they already have that constituent phenotype that help them to adapt to those regions. That's a very interesting question, and it's like a very big topic. And yeah, we would like to explore that area too.
Hi, thanks for the presentation. I think just thinking a little bit what you were saying, for plasticity for us, for breeders, is more in the sense of G by E. You know, we wouldn't like to have plasticity if you have a phenotype that is expressed. Like if you have an ideal phenotype, like let's say grain yield. So you can say it's plastic because it changes, but we would like to have the same grain yield. In that sense, we won't like to have plasticity. But I understand that there must be a lot of G by E. I mean, we're just thinking of soil. You can have different textures and structures of the soil that can complicate the penetration of the roots towards deeper layers. That can just simply make the phenotype expressed or not. But then I was thinking, have you thought of or like going on the opposite side, like taking genotypes that are good under stress conditions, and then see what's the phenotype in the roots. Like I can say, let's check what are the most drought tolerant varieties or the most nitrogen use efficient varieties, and then see what is their phenotype in the roots. So have you thought, or maybe you have those plans or something?
Yeah. We have thought about that. Well, I mean, not myself in the lab, because there is a paper about that from Stephanie Klein, and what she did was grow like a diversity panel of maize, and then she clustered like, okay, these are the good performance phenotypes, and these are the worst performers, best performers and worst performers. And then she kind of did a cluster analysis in terms of what phenes are. And basically what she found was that there is no one way to adapt to drought in terms of root. There are like many combination of phenes that give integrated phenotypes, and particular integrated phenotypes will give this adaptation to drought. That's one work available. But yeah, I mean, this is a good approach, because you are just targeting what will be the available integrated phenotypes there. But there is another possibility where there are maybe phenotypes that don't exist in reality, and they could be useful for drought, and we could select for those phenotypes. So with the modeling part, this is the thing that we can just simulate what would be like the combination of everything against everything, you know, and we can test if there are like maybe artificial phenotypes that could be useful under drought, and maybe if we found some good candidates, we can start selecting for those. That's for that. And for plasticity, as we were discussing before, maybe if you have a control environment, plasticity will be not required at all. We don't want plasticity, we have a constant environment. But if we think in, I don't know, low input conditions where there are no fertilizer or no water, and then the environment is very dynamic, so maybe plastic phenotypes could help. I mean, there is a huge discussion about if plasticity is useful or not. And I think, again, will depend on the environment you are growing the plants. For high input environments, I agree with you that plasticity is not necessary. We don't want plasticity there. But maybe for other conditions, it could be useful.
I guess all these things goes back to whether you have a specific adaptation of plant genotypes or genotypes that are wider adapted to different conditions. So it's I guess that's a never-ending discussion, I would say. But then when you say artificial phenotypes, then the question is whether those artificial phenotypes can be created. But if they are in nature and they provide advantages for the plant fitness, they will naturally be selected.
Yeah.
So it's a hard one, I would say, but it's very interesting.
I think we have a question online.
Yeah, I have one question.
Go ahead, Matthew.
Thank you very much for the presentation. Yeah, it's a very good work. But to be utilized by the breeder, actually, the most important thing is a very cost-effective phenotyping mechanism, like with regard to the number and the size of the cells and the like. Otherwise, we have to go for like molecular marker-based selection if it is associated with some markers. Have you ever thought of these things? Probably it may be a little bit early, but what concerns me is actually how to phenotype and identify a large number of population when we are breeding for these traits. Thank you.
Hi, Matthew. Thank you. Sorry, the audio here is kind of low, so I cannot listen very well. Could you type your question in the chat so I can read it and I can answer?
Can Can I repeat it again?
Just the sound is not good here. It's not helping us to understand.
Okay, I'll try to type it. Okay.
If you type it, I can read it loud. Let's try with Matthew's question. Let's see if we can hear Matthew better.
Yeah, I was wondering. Thanks for the talk. I was wondering, what do we know at the moment about energy costs? Can you hear me?
Yeah, I can hear you. Yeah.
Yeah. Because we have the carbon energy cost, the structural energy cost, obviously, of different routes. We've heard of steep, deep, cheap. What about all the costs associated with exudates, interaction with the microbiome? I mean, where are we on that? These seem to me like important things that black boxes, but could have very big implications on how we understand radiation use efficiency in general.
Yes, that's a very, definitely the environment is way complex than we can simulate until now, because, for example, in the model here, we can simulate exudates, but we don't simulate what will be the interaction of exudates and, for example, interaction with mycorrhizae or other microorganisms that will help to fix some nutrients. So I think, for example, I hear that Rudy Sowers at Penn State, he's trying to build a model of mycorrhizae and what would be the effect of mycorrhizae in the uptake of phosphorus and nitrogen. And we are thinking in simulating that. That's like a project that is there, but we haven't developed yet. And in the terms of radial, light intensity or the effects of light in terms of that, we just developed a drought model in OpenSIMRoot that considers the radiation, also stomatoconductance and leaf temperature. I was discussing that with Francisco too. And based on the leaf temperature, we are able to predict what will be the root phenotype as well. So we are exploring that area. That's very recent. That model was published last year. But I would say, Matthew, that we are working on that. We are, looking further that direction, but we haven't developed those models yet.
Okay, yeah, no, that makes sense. I mean, just as an example, an anecdote, let's say, Hemmer's work was leading work looking at radiation use efficiency at different growth stages, and found that there was a big interaction among growth stage with genotype. And of course, when we know nothing about the carbon cost of the roots, we can't really be sure that that's a true interaction or hidden aspects of carbon that we're not considering when we measure radiation use efficiency. So that's really, it's not just about the fact that the roots supply the leaves with necessary nutrients to enable a good expression of radiation use efficiency. It's actually knowing what is the total biomass that's being used underground. So I hope that area progresses. And thanks for the answer.
Thank you. Thank you.
Here I have the previous question. Is there a cheap and reliable phenotyping technique for screening the root anatomy in large number of segregating population?
The root anatomy. I will say that the most updated approach to quantify that is what we're gonna do in Sonora, is the soil coring and lacella ablation tomography on those. Still have some limitations that whenever we sample, we are looking for axial roots. That's the goal. However, if you are not looking at the whole root system, it could get some bias. Still, I would say that that's the best approach we have now. We are thinking how to perform better phenotyping techniques.
Okay. We should be closing this session. Thank you very much, Ivan. Give him an applause again.